Assalamualaikum and hello to everyone. My name is Muhammad Yazid S54420. Today I'm going to present of my proposal, which is the effect of temperature on the growth of the lobster Panulirus ornatus in Sabah, Malaysia, for subject MMB3005 Scientific Writing and Communication in Marine Biology. Firstly, the introduction for Panulirus ornatus. As you can see, there's the image of this lobster. They are also known as the spiny rock lobster, which abundantly found in Sabah, Malaysia. They live in the coral reef area under the rocks. Panulirus species normally exhibit 5 primary phases during their life cycle, which are adult, egg, larval stage, post-larval stage, and juvenile. Their body length can reach to 50 cm, but normally it is lesser, which is 30 to 35 cm. This type of crustacean has such as this tropical rock lobster have a hard outer shell or cuticle which serve as an exoskeleton or organic armor. And you can see from the box below the image is the taxonomy of this species. Second is the research background for my case study. Firstly, the temperature is one of the major environmental factors affecting the growth of crustacean. Second, growth rate has been shown to increase with increasing temperature to a maximum point before declining near the upper thermal limits. And lastly, lobsters are known to be thermoconformers or poikilotherms, and such their growth are determined with the environmental temperature. As you can see from the graph, the molting rate of the lobster increasing with the increasing temperature, but at one point they will start to drop when the temperature has become too hot for them. The reason why this study is very important because interest in the development of aquaculture of the tropical spiny lobster Panulirus ornatus has increased markedly over the past 10 years because of strong market demand and high price. Second, the market specifically seeks P. ornatus because of its larger size, vibrant shell color, and the purely luster-firm texture and sweet taste of the flesh. And third, P. ornatus are also important species in Malaysia in the coral reef area, so that's why this research will also fulfill the knowledge gap. The objective of this research is to study at what level of the temperature will affect the lobster growth of P. ornatus, and second, what is the optimum water quality for lobster growth whether in the wild or in the hatchery. Next is the instrument that will be used in the laboratory procedure. First, we'll have the fluorescent light for the 12-hour and 12-night day, and then red ammonia test kit, HANA pH and temp probe meter, and next is the Denver instrument for electrical balance. After that is the HANA DO meter for dissolved oxygen. Next is the titanium heaters for controlling the temperature of the water. And lastly will be the vernier caliper to measure the lobster carapace length. After that, we move to our study site. The study site will be at Double Bay, Sabah, Malaysia. It ranged from Tawau to Sempurna. This area was suitable because of the water is shallow with average depth ranging between 5 to 10 fathoms, which are suitable for this animal to grow. And there's a lot of coral reef there. Next is the method for sampling and laboratory procedure. The sampling of P. ornatus will be conducted at the Double Bay and will peak by scuba diving. After that, we will acclimate it in captivity for 1 month. Next, selected lobster will be stocked into the experiment system, and there they will be acclimated another for 4 weeks to the new temperature that we have designed. The water temperature and pH level were recorded 2 times per week, The wet week and carapace length was measured for every 30 days interval. This experiment took 121 days to be completed. Next is the data analysis procedure. The growth rate analysis will be calculated with their parent food intake calculation. Tested for homogeneity of variance and normal distribution using Levin test and Shapiro-Wilk-W test. The Tukey HSD post hoc test is used to assess the difference in final condition factor. A non-parametric Kruskal-Wallis ANOVA by rank test was used when the variance remained heterogeneous. A linear regression was employed to model the relationship between water temperature and the apparent food intake. This is the Gantt chart of my study case. The literature review, the proposal presentation, proposal writing, proposal submission, sampling, lab analysis, data analysis, thesis writing, progress presentation, final presentation, and thesis submission. And this, for the last page, this is the few reference that I have used to make this presentation and for my case study. And thank you guys for watching. That would be all. Sorry for any bad mistake.